Hello everyone and welcome to our module on cellular injury. Broadly speaking, there are four general mechanisms by which cells can become injured. The first one is if the capacity for adaptation is exceeded. In another video, I talk about cellular adaptation and how cells adapt to accommodate stressors in the environment. But that adaptation has a limit, and if that limit is exceeded, the cell can become injured. Another way cells are injured is by exposure to certain agents that are toxic or injurious. Cells can become injured if they're deprived of nutrients, and they can also become injured if there is a genetic mutation that leads to disruption of cellular metabolism. So all this is fairly obvious, but I just want to point out that these are the general ways by which cells are injured. Now, cellular injury is usually reversible up to a point, but if the cause of the injury is severe or if it persists, the injury may become irreversible, and at that point, this can lead to cell death. There are two general ways by which cells die. The first way is called necrosis. In necrosis, a cell essentially falls apart and spills its contents, which triggers inflammation, and the inflammation itself can damage more cells. Necrosis almost never happens to one cell all by itself. It usually happens to sheets of cells or tissue beds that are all connected together. The second way that cells die is called apoptosis. This is also called programmed cell death. This is a way by which an individual cell basically kills itself in an orderly manner so that it dies and does not cause harm to the body. This is a non-inflammatory process, and I talk about apoptosis in another video all by itself. What we're going to focus on here is the fact that when cells undergo death by necrosis, the cell death is preceded by classic cellular changes, and you need to know these for step one. These changes happen to any cell in the body that's going to undergo cell death by necrosis. For example, myocardial cells after infarction, or pancreatic cells in the setting of pancreatitis. These cells will go through some reversible changes of cell injury, and then if the stressor persists, the changes will become irreversible and the cell will progress to go through necrosis. First, let's talk about what happens to cells when they undergo reversible cell injury. So, for example, when myocardial cells are deprived of oxygen due to ischemia, initially they undergo reversible cell injury. If the ischemia persists or it's severe, then the cell will progress to die by necrosis, and we'll talk about what those changes look like later. But first, let's talk about the changes of reversible cell injury. And one of the hallmarks of reversible cell injury is that the cell will swell. This is a major feature of most forms of reversible injury. It is sometimes called hydropic change, which means water accumulation inside the cell. This is very difficult to see under the microscope. It's unlikely you'll be asked to recognize it on your exam. You just need to know that this occurs as part of reversible cell injury. And the reason it occurs is because when cells are injured, there's less activity of sodium-potassium ATPase pumps. Let's look at my drawing I've placed on the screen here. In many cells in our body, you find sodium-potassium pumps that are driven by ATP. So ATP is used to bring potassium into the cell and pump sodium out of the cell. If these pumps become less active due to injury, what will happen is more sodium will stay inside the cell. That sodium will draw in water, and hence you will get cellular swelling. In addition, sodium is often exchanged for calcium through a sodium-calcium exchanger. So when there's more sodium inside the cell, there's less exchange via this pump, and that leads to more calcium inside the cell. So the bottom line here is that during injury, when there's less activity of this pump, you end up with more sodium and calcium inside the cell, and that leads to swelling of the cell, one of the hallmarks of reversible cell injury. Another form of reversible cell injury is called fatty change. This is seen only in parts of the body that heavily metabolize fatty acids. So for example, you can see fatty change in the liver. You can also see it in muscle tissues like the heart and skeletal muscle. And in contrast to cellular swelling, this is very easy to recognize under the microscope, and you should know what it looks like. The cells develop lipid vacuoles in the cytoplasm, and this is a picture on the screen of a liver that's undergone fatty change. This is another example of reversible cell injury. If the cause of cell injury is withdrawn, the fatty change can go away and the liver cells can go back to appearing normal. Some other features of reversible cell injury are shown on this slide. First of all, the mitochondria inside of cells can swell. Remember, mitochondria are sort of like their own little cell inside another cell. So just like the cell itself can undergo injury, the mitochondria inside the cell can undergo injury. Membrane blebbing is another hallmark of reversible cell injury. This is a drawing of it on the screen. Basically what happens is there's a cytoskeleton inside the cell that maintains the cell in its normal shape, for example, circular. When that cytoskeleton is injured, then the cell loses its shape and pieces of the membrane start to form blebs. And then finally, you should know that cells that undergo reversible injury have dilation and enlargement of the endoplasmic reticulum. This is where proteins are synthesized inside the cells. And when there is cellular injury, ribosomes detach from the endoplasmic reticulum and there is less protein synthesis. You will read in your textbooks that reversible cell injury involves polysomal detachment from the endoplasmic reticulum. A polysome is a cluster of ribosomes. So what they're describing is this process of dysfunction of the endoplasmic reticulum, another classic feature of reversible cell injury. So now that we've talked about the changes you see in cells that undergo reversible cell injury, let's talk about the changes you see in cells that develop irreversible cell injury. These are cells that have progressed through reversible changes and are going to undergo necrosis. So once again, let's go back to our example of the heart when it suffers from ischemia. Initially, the myocytes may go through reversible changes, but if the ischemia persists, the cell is then going to enter the necrotic phase and undergo irreversible cell injury. So let's talk about what that looks like. Well, first of all, in irreversible cell injury, the membrane becomes damaged and the contents leak out, and this causes inflammation. And that's why necrosis is the form of cell death that is associated with inflammation. This is also very important clinically because when contents of cells spill out, you can detect them in the serum. And this is why we can measure the troponin level in patients with myocardial infarction or the lipase level in patients who have pancreatitis. Cells are undergoing necrosis and irreversible injury, and these things are spilling out into the serum. Another thing that happens when membrane damage occurs as part of irreversible cell injury is that enzymes called calcium-dependent phospholipases become active, and they further damage the cell. I'm going to talk more about this in just a couple of slides. Another feature of irreversible cell injury is that the lysosomes inside the cell rupture and they spill their contents. And as you know, lysosomes contain enzymes that break down other substances in the cell, so this leads to more cell damage. There are three classic nuclear changes that you can see in the nucleus of a cell when it undergoes irreversible cell injury. The first is called karyolysis. This is loss of the basophilic or dark color of the nucleus. This is a drawing of it on the screen here. These changes are very hard to see under the microscope, and it's unlikely they'll show them to you in an actual tissue sample on the board. You just need to know what they are. The second change is called pycnosis. This is nuclear shrinkage. This is also a picture of it on the screen here. And then finally, karyohexis is a fragmented nucleus. Basically, the nucleus falls apart, and then you see something that looks like this on the right side of the screen. Okay, so now we've talked about the changes you can see in cells when they undergo reversible injury. We've also talked about the changes you can see in cells undergoing irreversible injury or necrosis. What I'm going to finish this video with is a discussion of the mechanisms by which cells get injured and undergo those reversible and irreversible changes. And there are basically four mechanisms by which cells are injured to lead to all these changes. The first one is ATP depletion, the second one is calcium, the third one is mitochondrial damage, and the final one is free radicals, which I'm going to talk about in a separate video all by itself. So ATP depletion causes many of the changes seen in cells. There are many reasons that a cell can be deprived of ATP. If the oxygen supply is cut off to a cell, this can lead to ATP depletion. This happens in the heart when there is ischemia. Mitochondria can become damaged, and when mitochondria are damaged, then the mitochondria cannot produce ATP. And then finally, there are some toxins that directly affect the production of ATP inside of cells. When ATP becomes depleted, the cell can't maintain its membrane pumps like we talked about earlier, and the cell can't synthesize proteins, and this leads to reversible and irreversible changes. It's high yield to know that calcium inside of cells is very bad, and when calcium gets inside of cells, this leads to cell injury. Normally, the calcium level is very low compared with outside the cell, so an influx of calcium is one of the hallmarks of cellular injury. Very high yield to remember this. There are a couple of reasons that calcium levels inside of cells can go up when they're subjected to a stressor. Calcium can be released from intracellular storage. It can also influx across the cell membrane when the membrane becomes damaged. Calcium causes cellular injury through a couple of mechanisms, and one of the main ones to know about is what I mentioned before. There are enzymes inside of cells called calcium-dependent phospholipases. These enzymes are activated by increased calcium, and they break down membrane phospholipids. That's why they're called phospholipases. So anytime calcium gets inside of cells, it will activate these enzymes, and the cell will basically begin chewing itself up. In addition, calcium inside of cells can also damage the mitochondria, which can further lead to cellular injury. Our final mechanism of cellular injury for this video is mitochondrial damage. So how can mitochondria inside of cells become damaged? Well, first of all, mitochondria need oxygen and energy to function just like the cell itself needs oxygen and energy. So if there isn't enough oxygen, the mitochondria won't be able to generate ATP and won't be able to maintain its membrane and structural proteins. Reactive oxygen species are a type of free radical, which I talk about in another video, and they can damage mitochondria. And then finally, mitochondria have something called the mitochondrial permeability transition pore. This is a hole in the mitochondria that is opened by calcium. And as I told you before, calcium influx causes lots of damage to cells. So not only can calcium damage the cell, it can also damage the mitochondria by opening this pore. This will cause the mitochondria to lose its membrane potential, and that will lead to failure of oxidative phosphorylation, and that will lead to less ATP for the mitochondria and for the cell itself. And that concludes our video on cellular injury.